Good evening, everyone. Both here in person in Parkville in Melbourne and also online. We're joined tonight for the Victorian Division seminar on biomanufacturing in Victoria. My name's Jan Tennant. I'm a member of the Victorian Division Committee, and it's my very great pleasure to welcome everyone to the event tonight. I also acknowledge the traditional custodians of country throughout Australia and their connections to land, sea and community. I pay my respects to their elders past and present. I'd also like to pay my condolences to the Alice Springs Indigenous community for the devastating events over the last week or so. The Victorian Division Committee is delighted to have invited you and to see you all in attendance tonight at this event presented by the Australian Academy of Technological Sciences and Engineering. Victoria has quite a strength in biomanufacturing, but we're often, not only in this state but in Australia, it's often said that our problem, the hard problem in fact, is in translation. So how do we go about turning promising laboratory processes into robust, compliant, commercially scalable manufacturing? We're fortunate this evening to have two terrific speakers lined up to talk about that particular topic in detail. And what I'm going to do now is actually introduce them both, describe their bios to you, and then I will hand over to the speakers directly for the rest of the evening, and we'll follow up their presentations with the traditional Q&A. So get your thinking caps on as you're hearing the presentations. Jot some notes down about the question that their commentaries have inspired in you. We will allow plenty of time at the end for those Q&As to come out. Our online attendees, you know the drill, I'm sure, but we have someone here in the room, Melike, who is actually going to be recording your questions, and she'll pass those on to me and I'll ask them on your behalf. So thank you for listening in from afar. So our speakers tonight, the first of whom is Dr. Brenton Hamdorf. Brenton is the Precinct Manager of Thermo Fischer Scientific, company that is regarded as the world leader in serving science, driven by a deep commitment to enable their customers to make the world a healthier, cleaner and safer place. Brenton is responsible for facilitating Thermo Fisher's interactions with organisations that are co-located within precincts and the realisation of significant collaborative opportunities. Brenton's extensive experience in commercialisation of early-stage technologies from across the life sciences, biotechnology, environmental and industrial sectors. He has driven and been responsible for the creation of 14 startups covering topics and innovations including tissue engineering, gene therapy, diagnostics, small molecules and peptide-based drugs. These and other opportunities have arisen from within universities, via clinicians, from cooperative research centres and from private entrepreneurs. And he's also created more than 15 collaborative partnerships and secured many millions of dollars in both research and collaborative funding. Brenton holds a Bachelor of Science in Microbiology and Biochemistry with honours in Molecular Biology. He has a doctorate in biochemistry and a master's in business and business administration with special focus on entrepreneurship, marketing and finance. Just to add to the whole picture, he is also a registered technology transfer professional with over 25 years experience. Brenton's going to be our first speaker tonight, and he's going to be followed by our second speaker, Dr Felicia Pradera. Felicia is Director of Moderna's Regional Research Centre and Strategic Alliances in Melbourne, where she is responsible for strengthening Australia's role in the global mRNA innovation system and for leading R&D strategy, partnerships and ecosystem development across Australia and the Indo-Pacific. With over 20 years' experience across government, industry and research, Felicia has made significant contributions to Australia's health, security and technological capability in medical countermeasures. While she was at the Defence Science and Technology Group, she established Australia's National Medical Countermeasures Program and led advanced development initiatives bridging research and deployment. Later, as chair of the Quadrilateral Consortium, Medical Countermeasures Consortium, Felicia brought together the defence and health departments across the United States, the United Kingdom, Canada and Australia to strengthen international collaboration. She has advised national and international governments on pandemic preparedness and response, including briefing the US National Security Council, and has served on the Australian Prime Minister's Science and Industry Technical Advisory Group during COVID-19. Felicia's experience, knowledge, and indeed her work at Moderna sits at the intersection of biotechnology, innovation, policy, and industrial capability, with a focus on accelerating the development of scientific advances and next generation medicines into scalable public benefit. Not to be outdone by Brenton's formidable series of qualifications, Felicia holds a doctorate in biotechnology, a Master of Intellectual Property Law and a Bachelor of Science with honours, and she's also a non-executive director of several small biotech-related organisations, companies. So I think you will appreciate, as have I, that these two presenters tonight have a wealth of experience and knowledge and are involved directly in, embedded deeply in biomanufacturing. And it's my pleasure to invite Brenton to come to the podium tonight first to present his work, and we'll follow on immediately into Felicia after Brenton. So welcome.
Thanks, Jan, and to the ATSI team for inviting me to speak today, and thank you everyone for giving up your evening to come and listen to Felicia and I give you a little bit of a background on some of the things that are happening in bioprocessing in Victoria. So without further ado, I guess quickly, a quick show of hands, who's involved in bioprocessing directly in the audience? People who, yeah, and who completely new to the bioprocessing area and just here for interest. Excellent. That's good, because I'm going to talk a bit of general and a little bit of detail as we go through. So hopefully nobody gets lost as we go through. So I'm going to focus on one of the challenges in translation. So really taking, I guess, new discoveries out of a research laboratory and then taking them into a manufacturing environment and what that entails. And some of the, I guess, challenges that individuals face as they go on that journey. And I've experienced a few myself, and I've seen quite a few as well. I'll go manual. Just a quick disclaimer. Oh, got that. Excellent. These are my opinions, not necessarily those of the company that I represent, but certainly some elements of Thermo Fisher are here. So as we look at the spectrum here of bioprocessing, it's actually quite diverse. And for those that aren't aware, I'm not going to touch on mRNA too much, but it's a relatively small platform in comparison, and I think Felicia will talk a little bit more about that. But I'm not going to focus on those. I'll focus on the other areas and leave it for Felicia to look at. Protein and antibody production is a pretty established process within bioprocessing at the moment, predominantly mammalian cell culture, and there are biosimilars. And so biosimilars in the antibody side of things are effectively the same molecule, but produced in a different organism or a different way or a slightly different process. And so although they're supposed to be the same thing, sometimes they're not quite the same. And so you'll find biosimilar products operate slightly different. Microbial fermentation really looks at much larger scale volumes of production. So this is 20,000 litre tanks or vats of microbial fermentation, typical for the brewing industry, for instance, but also for the production of other molecules of interest that are produced through microbial fermentation. It's a pretty robust process for the microbial side of things. It's pretty well known. but obviously there are lots of challenges still in the scale of production as you increase things. AAV gene therapies, probably the most complex drug you can make. It was one of the companies that I was involved in setting up, was an AAV gene therapy company. Really difficult to manage yields, really difficult to manage the products. Quality, chemistry, manufacturing and controls processes generally hold up the approval of drugs through the AAV sector quite frequently. Lots of new combinations coming out though. So there are different types of AAVs and different inserted genes that they're putting in, and whether it's RNA or DNA, it doesn't really matter. And the last one's cell therapies, and cell therapies have gone through a bit of a cycle. They were popular a little while ago, they disappeared, and now they've come back again as CAR Ts have emerged as the dominant cell therapy that's coming out. Lots of hospitals are running and producing CAR Ts, so it's like mRNA, it's actually a relatively small production suite, and there are some constraints, and also some relaxed rules which allow them to be produced in hospitals in small batches. So they're quite different, but they all have different elements to them and different processes. So I'll look through a few different things. We'll talk about the tech transfer piece, some of the regulatory issues, analytical components that are necessary to manage the production process, and finally a little bit about workforce development, which is one of the challenges that's emerging across the scene for how do you staff and actually grow this market if you can't have the right workforce. So a little bit on just some market data so you've got a little bit of context. The overall pipeline, this was in Q4 of 2025, 2,041 drugs across gene, cell and RNA therapies that were in development, but it was lower than in the first quarter of last year. So the actual number have dropped in terms of new products coming through. mRNA is still the dominant RNA modality that's coming through, although Felicia might have some updates on other things that are coming through that pipeline. And cell therapies are actually one of the most active, in part because some of it initially is very simple to do. So there were 70 approved and 875 in the pipeline coming through. This is in the US data. That just gives you a little bit of context. So why is translation the hard part? And it's not necessarily hard. The process is there from research, process development, the GMP transfer, and then clinical and commercial expansion or sale of products. But in between those steps, there's a lot of challenges. And the first one is technology transfer, as we go from the research environment into a commercial environment. We work on that process development. And then as you go from the process development into some sort of GMP transfer, there's all of the CMC and regulatory issues that have to be looked at. And as you go through that process, you've also got to develop all your analytical steps. And lastly, it's around the workforce that I mentioned before. So these are what I've called the bridges, and bridges that lead to or enable technology transfer to occur. So I spoke to our head of production at Patheon in Brisbane, and she said a couple of things. One is start with the end in mind when you're designing your process. You have to know where you've got to get to, and then move that through and consider all the things that are required to get to that end point. And the other was that not everything can be scaled. So you can see here there's a scale-up staircase here as you increase in volume as you get to more commercial production. And so as a research lab, you're dealing with 5 or 10 litre volumes at most, typically. And as you go through the process, you can end up with 2,000 litre reactors, or higher if you're talking about microbial systems. But as you go through that process, you end up with this whole range of different issues that come into play. And so I've listed a few here of mixing. How does these volumes mix? How does the viscosity change over time? Oxygen transfer through larger volumes, particularly if you've got a very dynamically growing culture. How do you get the oxygen to all of those cells equally? Shear as you begin to mix and stir these large volumes. And then how long are they in there for and exposed to or able to produce their products? What's the residence time in the reactor? Then you've got considerations like in engineering scale, whether you're using a single-use technology or a stainless steel clean-in-place type technology. Most laboratories deal with glassware and reactors, which are 2.5 litre flask in a shaker flask. We're talking here very large vessels or sealed plastic bags. And in fact, the single-use is probably the dominant modality at the moment for a lot of the everything up to around 2,000 litres is generally done in a single-use format. Different types of media, the buffers that go into these, and the supply chain, the sourcing of those materials. As you increase the scale of everything you're doing, all of these things have an impact. And then as to maintain efficiency of your system, automation comes into consideration. You've got to understand the data integrity and the batch record discipline. As you're producing these products, you need to record all of this information. And why it matters is because you're not just transferring a technology in terms of the science, you've actually got to transfer the process. And that process has a lot of engineering in it, a lot of data transfer, and a lot of, I guess, quality elements that have to go in to manufacture this drug, not just once, but 100 times over exactly the same, and that's the challenge. The earlier you design that manufacturing process, so in that tech transfer part, the earlier you can think about what has to be done, the easier it is later on. If you leave things to the last minute, or you think, I'll deal with that later, it comes back to bite you because you've missed a step, or you've got to go back and redesign things to actually deal with the steps that you've missed. And then Thermo Fisher really does offer a range of bench to certified GMP type products and services that do de-risk your production process over time. So we know that we can take something from a lab bench and actually put it into commercial production, and we've validated that process previously. Second one is regulation and compliance. It changes across the board as soon as you start going into production. And depending on what you're producing, that pathway difference. So here I've done just the simple one of a single small-scale cell therapy treatment that is done in a hospital setting under a regulatory, loophole, which allows you to do a first-in-human study with just using a clean room conditions. But the problem is, as soon as you decide you want to, there are some requirements here for doing that, but as soon as you want to then go into broader use of this, you must be compliant with full CMC and manufacturing requirements for commercial scale. And so the bar gets lifted. And so what they've found is a lot of people will do their first in human study and it will never progress to wider adoption. So there's lots of drugs that, particularly cell therapies, that never progress any further because the hurdle here is quite large. As you go through here and looking at your drug registration process, what is it? Is this a over-the-counter drug? Is it a prescription drug? Is it some sort of a mechanism for delivering your drug? Are we taking which pathway using? Normally for drugs, you want to follow the registered drug pathway. And so certain compliance requirements are needed. And if you're doing some of the more complex drugs, is anybody here from TGA? When I was doing the AAV development, we were really relying on FDA to be able to provide us with the guidance necessary for that development, because it was so complex and there were so many elements that arguably the TGA hadn't had enough experience or exposure to dealing with these drugs. Quality controls and evidence, and obviously your eventual CMC package that goes with your regulatory dossier. The bottom line really is our first in human trials are relatively simple, but as you scale up and you want to go into a broader population, particularly with the cell therapies, and this I'm just using cell therapies as a reference here, you need to lift the bar, and that bar is quite high for those drugs. Third one is the analytical piece. And so for a lot of these processes, the bioprocess steps, as some of you are aware, at every stage in that process, you need an analytical test to validate all of these elements. Do you have the right molecule? So if we're talking even about the raw materials that are being fed into our production line, do we have the right material? Is it the right strength material? Is it pure? Is there any contaminants, other materials in there, other bacterial species or something else? Is it safe? Does it have endotoxins in it? And so you have to screen your raw materials, and then every step of that process also needs an analytical test. And so quite often as people come out of the laboratory, they've developed their product and it worked, cured a few more mice, But when you go into production, if you don't have all of those elements in place, it will delay you because you actually have to go back and develop those assays at every step because you have to validate every single step in that production process. And so this is here's the credibility behind that system, because you have to be able to do this every time, 100 times over, and produce exactly the same product. And as I said before, AAV was one that I was working on. Lots of different assessments in that translation process. Empty versus full capsids. Okay? Seems relatively simple, but it's actually not, because you have empty, partially full, and full capsids. And even when they're full, does it actually have the right thing in them? Does it have the gene of interest, or has it got something else packaged? So empty versus full is not a straightforward question. What other proteins have been encapsulated? So as this protein forms, this AAV capsid forms around your protein or your DNA of interest, what else is it encapsulated? So you've got to understand those. Is it the right thing that you put in there in terms of the genome? Has it been modified in that process of production? And then you've got to be able to say that if I produce this batch today, if I produce another batch in a year's time, is it actually the same thing? Because I'm only approved to produce that batch. So this is where all of the analytical quality control systems really come into their own. And workforce capability. One of the challenges is, as this sector grows, there's a need for people to come into the sector that are experienced in dealing with this highly regulated drug production environment. A lot of people graduate out of a university with a Bachelor of Science, honours, or a PhD in something, but they haven't worked in a regulated environment. And so there's a need for these people to be trained up. So if they get employed by a Moderna or a Pfizer or a GSK, they then need to be trained up on how to work in those environments. Now, where do they get that done? I'm running a production site here where I'm producing drug product worth millions of dollars for a customer or for ourselves or for patients. Can I afford to train people in that space? It's actually really hard. You can do 1-to-1, but you can't do 1-to-20. It's just not feasible. You cannot put that many people and train them. So you need a space in which to train them. And so researchers know the science, but they don't necessarily understand this regulatory environment. Industry needs people who are GMP ready, and universities continue to produce the talent. But what was needed is the simulation labs and the ability to partnership training and vendor supported capability building that widens the capability in the state, so that we don't have to get people from overseas to come and do the work that could be done locally. And you can't just learn it from reading the SOPs. It's an actual exposure. You've got to be in these things from gowned up from head to toe, and it's a process just to get into them. So it's not at all straightforward. So why can Victoria lead in this translation story? We're a big market, $21 billion for the health technology sector. So this is out of the Invest Victoria report, 2025. It's $3.8 billion worth of exports out of Victoria in the space, and 51,000 people employed across the sector. So we account for more than half of the pharmaceutical exports that happen in the country. So that's pretty impressive. Opened the first accredited manufacturing for targeted cancer therapies. Seqirus operates the only cell-based influenza therapy, and we've got the National RNA Blueprint Capability Network based here as well. So the opportunity here is really to lead not necessarily every modality, but solving some of those gaps that exist that enable others to produce what they need to produce. And those bridges kind of point to some of those areas in which we can do some work. And so what can government and ATSI potentially could do? Look at shared translational infrastructure, and how does that allow people to scale and pilot things, their processes, and make them more robust. Support on the regulatory piece. You don't know what you don't know until you get into these things. It's quite difficult to understand what you need to know unless you're paying a lot of money for consultants to come and tell you. So facilitating and directing and helping people along that path is really important. Some sort of workforce academy model where we're sort of creating a simulation training, creating operator pathways and letting people understand how do you work in these environments? What do you need to know to do it effectively? And last one around APAC, partnerships, stretching that out, being a regional hub for these types of capabilities. and supporting others to do their translation and drug development here locally. So AXI can add value as a convener of industry, university, regulators and government, focused on those translational bottlenecks that no one player can really solve by themselves. And I guess in closing, 3 takeaways. Bioprocessing translation is where the scientific discoveries meet commercial reality. These bridges of scale-up, compliance, analytics and workforce capability are essential to developing that bioproduction capability. And Victoria can really strengthen its resilience by treating translation as shared infrastructure and not as an afterthought. So it's part of an integrated process, not just part of it. But so as Thermo Fisher, we do deal with equipment, media, analytical QA. We've got Patheon Business, which produces its own drug product in Brisbane. And then training and collaboration. We do all of those things. We're part of the ecosystem, but we are not that ecosystem. We need everybody else. For Thermo Fisher to succeed, we need others to succeed as well. And so that's why we're supporting the growth of, bioproduction capabilities across the state. We have a strong interest in making things happen and helping our customers and new and emerging customers be successful in what they're doing in the bioprocessing area. So, thank you. With that, now that I've warmed everybody up, I'll hand over to the main item of tonight. I'll hand over to Felicia. Thank you.
So, thank you very much for inviting me to come and speak today, and I'd like to say thank you to Brenton because I think he's really given a great overview of bioprocessing in general. My remit's really about the Moderna story and how Moderna has come here to Victoria, to Australia, and the work that we're doing in building up the Australian and Victorian ecosystem. So I'd like to start by, because we're talking about bioprocessing, acknowledge the traditional lands on which our facility is built, and that is the Boronong people of the Kulin Nation, and pay my respects to the elders past and present. So many of you may know that our site's actually out at Clayton at Monash, and I think what's really fascinating about the site, which we have established our manufacturing facility during COVID-19, it was a testing clinic. It was a big empty car park that people drove around. So now we have a manufacturing site, which is right there. And prior to that, it used to be a drive-in movie theatre. So there's quite a bit of history in that location. And it's a really important location, being co-located with the Monash precinct. And of course, we have our regional research centre here in Collins Street, and we work really closely with the Parkville precinct and the amazing academics and scientists that are across not only Victoria, but of course across the country. So Moderna is actually really unique when it comes to a large biotech, and I'm really conscious to say we're a large biotech, not a pharmaceutical company, because we're around 4,200 people, and we focus solely on mRNA. It is the only modality, and we use it as a platform capability across multiple different areas, and I'll get in a little bit later into the different therapeutic areas that we're engaged in. And so everything we do is based on that platform and that platform capability. And so for those of you who are aware or aren't aware of what mRNA is and how it works, it's really a messenger. So it's messenger RNA. And what it does is it instructs your cells to produce specific proteins. And that, if you think about it like as in software code, it's a piece of that software code that allows execution of a particular sort of set of information that enables you to do an activity. And so it's really, really sort of potent in how we're able to instruct the body to respond to an infectious disease or to cancers or to rare diseases. But mRNA in itself needs protection, and you'll have heard about lipid nanoparticles because it degrades very quickly. And so lipid nanoparticles are a key part of that platform capability in order to protect it, to deliver it to cells and enable it to act. So I think for a lot of people, they only really heard about Moderna during COVID-19 and are not really aware that there has been a long history of development. And so the organisation really was stood up in 2010 and it came out of Cambridge, Boston and MIT and Harvard and the sort of collective smarts of academics there. We've, actively worked in the beginning around sort of influenza vaccines, sort of around 2015 and 2017, the publication of our sort of first lot of work in mechanisms of action and how it worked. And then, of course, publications associated with LNPs and how they were developed and integrated. And then, of course, during the pandemic, this is where all of that lead-up information was able to be sort of collated collectively, rapidly and effectively to develop the SARS-CoV vaccine. Now, of course, it was really critical, all of the research and development, the clinical trials, the regulatory, the CMC, which we've been hearing about, and the production, all met the appropriate regulatory standards, not only in the United States, but also across the globe to enable the vaccines to be delivered to the population. And so we have ongoing programs, broad programs across respiratory viruses, latent viruses, oncology, and other sorts of rare diseases. One of the other things that we do is because we have a platform capability, and of course there are many infectious diseases that we don't have the researchers in-house to be able to do the work. We do a collaboration essentially with academics across the globe, and this is through our mRNA Access program. So there are around 140 different pathogens which are on a list, and any researcher that's doing any sort of work in that particular space can apply to Moderna. And we will provide mRNA and mRNA LNP free of charge for them to do some preclinical experimentation to see if their particular targets show any promise. And so we work collaboratively with them because it enables us to see where potentially our future pipeline and collaborations could occur. So let's talk about Australia and let's talk about Victoria. So the onshoring of capability really came about as we're aware that, sovereign capability during the COVID pandemic was really highlighted. And so the Australian government indicated that they'd like to have mRNA capability brought to the country to build the appropriate supply chain so that we wouldn't be put in the same position as we were during the pandemic. The Victorian government as well, they also had a strategy about mRNA, and some of you may have heard about mRNA Victoria. That is a standalone agency within their sort of government department. It's the only one that we're aware of globally where the Victorian government thought that mRNA was a capability that was essential to bring to Victoria, that they stood up an agency to enable those relationships to be built. And mRNA Victoria, of course, we're also very successful in bringing our competitors, or as I like to say, our co-opetition, BioNTech, also to Victoria. It is the only place in the globe where both large mRNA capability companies are, down the road from each other. So it's quite a significant insight that the Victorian government had to bring both mRNA capabilities to Victoria, but also in building that capability more broadly for Australia. So our relationship, of course, is not only around respiratory medicines, but also pandemic preparedness and preparing us for the next pandemic. And so the intent is to reduce that supply, that sort of dependence on global supply chains, onshoring capability, onshoring workforce, and the training of the next generation. And my role really is about building that ecosystem and the research and development that will be part of the pipeline that goes into producing, what might be our next sort of lot of vaccines or therapies. So I'll flick to the next slide. Oh, this is a build. All right. So for our big moments, and what is really quite remarkable, so in 2022 was when the contract was signed with the Commonwealth and Victoria, and we broke ground. Now, we are now 4 years later. The facility is an end-to-end drug substance and drug product facility. We are sort of fully accredited, so our licenses, and we'll have the first lot of Australian-made product coming off the run at the end of this year. So 4 years for an entire facility to be built is quite remarkable. There has been an enormous amount of work that has gone on and continues to go on at our manufacturing site, and the team's working, long hours for both drug substance and drug product. And I think it's really important, and for those of you who don't know, so drug substance is the content that goes in, drug product is for the fill and finish. So sometimes that goes into vials and sometimes it goes into injections, plunge syringes, thank you, brain. So this requires different branches of in the manufacturing facility and different accreditations that are required and different SOPs and different trained people, and so very distinct. What's unique about the Australian facility is it's actually the only current end-to-end facility in the Moderna network. So, and all of our other manufacturing sites are drug substance, and then we partner with a drug product CDMO. So in the intent of the Australian facility is during pandemic, we can produce around 100 million doses of vaccine now, which is way more than, of course, the Australian population would require. But where are we in Asia and the Indo-Pacific? And the Australian government wanted to make sure that they had the opportunity to donate to our neighbours in our region, should that be necessary. And at the moment, we have around 140 highly skilled staff. So 110 of those sit at the facility, and then we have 30 here at headquarters in the Regional Research Centre. We have 80% of the construction materials were sourced here locally and manufactured in Australia. So, we're very proud of the site. So part of our contract and arrangement with the government is really set around 3 pillars. Manufacturing, of course, critically important, but also the inputs of research and development and workforce and supply training. And I have the great privilege of leading our research and development components. The original focus, of course, was in infectious diseases because we had just gone through a pandemic, so respiratory infections and infectious diseases. But as we've seen, there is great application in oncology for mRNA and mRNA-based technologies. And so we're doing a lot of work in the ecosystem and mapping really where that capability can be integrated into our global R&D programs. So this just gives you a picture of the scale which Brenton was talking about in relation to the standard industry biotech versus commercial manufacture for mRNA. So our footprint is significantly smaller. So commercial manufacture is anywhere between 30 to 100 litres in that. And then, of course, you've got the clinical manufacturing capability. So it's cell-free, so contamination risks are significantly reduced. We have flexible, non-product dedicated plans, and of course it's small footprint. And as Brenton had sort of mentioned, things are done in bags, they can be taken out. The processes, because we're using a platform capability, all we have to change is that sequence that goes into our LNP for a new disease. So it's a really sort of interchangeable capability. So this just gives you an idea of what our filling line looks like and the sort of elements associated with our drug product production. So all in sort of contained and safe environment. And it's one of those things with drug production, with drug product, when we were looking to establish the facility here, we thought, oh, well, we do drug substance, absolutely, and then we do with a CDMO. But when we looked across the country to partner, there was nobody who could do drug product. And so therefore we've had to sort of build that capability. But what that has meant is that we're training up the next generation of drug product specialists. We had to bring in people from overseas, but at the same time upskill capability. And so now, you know, as I said, we've got a really capable workforce. They're also interchanging between drug substance lines and drug product. So that's sort of model I talked about around sovereign capability. What was really interesting at the same time as Australia was looking to bring onshore manufacturing, so was the UK, so was Canada, and I do love it's all Commonwealth countries. And so what we've formed really is what we call the Resilience Network across the US, UK, Canada and Australia. And we work really closely when it comes to sort of that sovereign capability, health and economic security. And I'm engaging constantly with my my counterparts from R&D because I'm trying to say that Australia is better for R&D. And we've got smarter people and innovative people. So it's always a really good sort of healthy, a bit of competition that goes on so we can put investment in the country. But as I mentioned, Australia's the only one currently with that drug product and that full end-to-end. But what we're seeing now is that we're also at Norwood in the United States, we're going to be putting in a drug product line. But the wonderful thing is the skilled people from here are going and teaching the people there. So it's that reverse sharing of information. And of course, there are emerging partnerships that are happening. Not only is there Mexico here, but we're also doing a few more in Southeast Asia. So this just gives you an overview of the pipeline. We have 22 assets here that are on our sort of targeted development programs. We have 44 in broader development across respiratory vaccines, latent, oncology and rare diseases. And so there's a lot of activity going on, and I'm really sort of excited to see where our oncology portfolio goes. And we're seeing some really great readouts at the moment from some of the phase 2, phase 3 data. So R&D activities, this just gives you a bit of a snapshot of the sorts of things that we're doing here in country and running clinical trials. And we have our accelerators in partnership with Monash University. And of course, workforce and supply chain, I cannot emphasise enough how important workforce is and the opportunities for training that next generation. So they are useful to the industrial partners and the biomanufacturing ecosystem that we have going on. I'd like to just sort of finalise by saying that, you know, we see a 3-part value system around the work that we're doing for each of these countries, and it's around sort of that security for Australia, growing that economic and industrial value, and of course, you know, uplifting that innovation as part of the global network. And finally, the Oxford Oxford Economics did an impact report of the establishment of our facility in Australia, and they said that it is estimated that it will bring around $4.8 billion savings over the sort of pandemic period of 30 years. And so when you think about the R&D benefits, you think about, you know, what that's going to mean for Victoria and for the country. And these are conservative figures. And so, you know, I think that it is an investment that both Moderna and the Commonwealth and Victorian governments really see the value for our biomanufacturing landscape. Great, thank you.
Thank you very much, Felicia, and also Brenton. Would you come up and sit together and, I'm going to say, make yourself comfortable for some questions? I'm sure there's a few brewing here in the room, and also possibly some that are coming in from our colleagues who are watching online. You've given us a lot of food for thought tonight, and very happy to open the floor to some questions.
Well, it sounds like a fantastic facility, but as we're hearing now about blockages of supply chains when events happen in the Middle East and so forth. I assume that your company's got everything worked out so you can keep working should some global disaster happen and we're isolated, as we've been before.
Yeah, so look, that's absolutely part of what we've been doing. So really over the last sort of 2 to 3 years is understanding what actually can be done here in Australia and who across the Australian ecosystem can support what we're doing. Where are there redundancies, and then where within our global networks across the UK, Canada and the United States can we get the things that are not possible. And so there are lots of plans and contingency plans that have been put in place to enable that to occur should a pandemic be called and we get shut down, the country gets shut down again. So yes, there's a lot of work and we have people dedicated to make sure that that doesn't happen. Arthur?
Thank you. Oh, hi. Look, it's lovely to hear about great manufacturing and manufacturing consistency and things like that. But the other side of the equation is how the body reads your modified RNA. And there's some work out of Cambridge University on frame shifting and the body producing off-target proteins. Now, I was looking at Claude today, and Claude says that Moderna are actually looking at codon sequences that are less likely to be misinterpreted to produce these off-target proteins, which are safe, apparently. Can you tell us a little bit about that and frame shifting and what Moderna's doing to essentially recreate millions of years of evolution to develop codon sequences that cannot be known to cause harm in the body?
So this is a highly technical question, which I'll be honest with you is not my area of expertise. I can take it on notice and I can get back to you on that. But what I will say is that the products that have currently been approved on market have met all of the regulatory requirements and standards, and there is, clinical evidence which has been gathered associated with their intended outcomes. And that they have, we very, very carefully monitor what happens when they are then on market with the any reports that come in. And so all I can say is that, from what we have seen, our vaccines are safe, they are effective, and they are very, very closely studied.
Paper I can show people.
Yeah, that would be lovely. Yeah, that would be great.
I'm going to chip in and ask a question. You've talked about regulatory environments, and I know we sort of touched on this before, but why do we have separate regulatory environment in Australia when surely this is now a global situation? Why do you guys have to go through Therapeutic Goods Australia, or whatever it stands for, but the A is Australia, versus a global situation?
Do you want to answer that, or do you want me to answer that? You want me to answer that one? Okay. All right. So every jurisdiction has its own regulatory authority. So of course it's the FDA in the United States, it's the Therapeutic Goods Administration here in Australia. You've got the European Medicines Agency, and then of course you've got when the UK did Brexit, they now have their own regulatory agency as well. I think, each nation has dictated legislation, and under their legislation, which they follow in their health requirements, they, have specific things that they need to look after. And there are nuances and differences in the way that they're run. So the FDA is a very different organisation to the TGA. It has scientists that are doing research work as well as the assessors, whereas the Therapeutic Goods Administration.
No, it's not Australia. That's
so the TGA, they rely heavily on the data packages that are coming in, and you have the people going through and analysing all of that information. So they have very similar principles, but there are nuances based on the requirements of each of those countries. And there's a lot of work that's going on at the moment around how to harmonise. And so learnings from different jurisdictions are shared, and they do talk to each other very often. But, you know, some things might go through Canada quicker than they will here at TGA. And so we just have to, you know, at this point in time, we have to deal with different regulators and where we're producing, you know, our different medical products.
Do you see, Felicia, a role for the academies in advocating for some harmonisation?
I think that doesn't hurt.
We've got a question. Oh, one here, Matthew first and then Margaret.
Yeah, I had a question for Brenton. Look, in your comments towards the end about how can government help and all that, you mentioned the idea of shared infrastructure that can be accessed by small companies and so on. I guess this is an idea that keeps coming up whenever we talk about advanced manufacturing. And it sounds particularly difficult in this area because the processes are all so difficult and complex. So have you got a specific idea in mind? Have you got a magic solution on this?
I do, don't you?
Well, I'd love to say yes. I do have a magic solution.
That's a wrong answer.
Look, I think there are a few examples of some shared facilities that exist. So one that's easily accessible here is cell therapies. So they've got a facility which is anyone can come in and use. There are a series of clean rooms and they're basically set up for companies to come in and do their drug production, or they can be done by cell therapies. The UK ran a program called the Catalyst program, and that was acquired, I think, from the closure of GSK or one of their production sites. Might have been AstraZeneca even. They closed a production site and the government acquired it, and then what they created around that was a series. So they had GMP production in the centre, and then around that they had the development labs that went and helped them optimise their production processes. And I think that was quite a good model. I don't know if they're still funding it or not, but it's certainly been going for a while. I quite like it in terms of a model for shared access, because running a GMP facility is really expensive. And you don't, unless you're producing drug substance or drug product, it's not worth running through a GMP facility. It's just that expensive just to operate, because you have to validate the site. As soon as you turn it on, you've just got to keep it going. So that sort of hub and spoke type model where you've got one centralised GMP facility supported by a range of development laboratories which may develop different modalities, I think works quite well. It'd be fantastic to see something like that established here. And then you would have to build into that workforce capability development. And it's one of the other areas of interest for myself, is how do you build that capability so that you don't have to reinvent the staff every time. Like there's a growing capacity, a capability, a resource that people can draw upon, whether they're setting a startup up or whether there's a new large biotech moving into town. And then people can move between them and the exchange of knowledge and information, and it gets to, I'm assuming, a critical mass where you've got enough capability in the market that companies can grow and shrink and people can move amongst them. But you've also got a training capability that continues to evolve as new technologies come out and people need to be retrained on new bioprocesses, new equipment that comes out, new modalities that get developed. So there's this constant need of training, whether you're already operating in a space, so your staff will need to be continually trained. They're not trained and they're done and dusted. There'll be something new that comes out and they have to go back and be trained. And that's difficult, as I said, to do in an operating GMP facility. It's just really difficult.
And I think the other element of that is around surge capacity. So if the proverbial does hit the fan, you are going to need more people to run more shifts. So how do you prepare them through something like that you can then sort of acquire them as you need? So,
Hopefully that answers the question.
I think it went a long way towards it.
This is a question for Brenton too. I was really interested in that marketing data you had from the US, where you showed that some products in bioprocessing had actually dropped in its capacity in the FDA. Is that because technology is changing and we're much more moving into more specialised mRNA, etc., or is it the FDA's sudden change in direction and that hardly anyone who used to know stuff is no longer there? It's very slow, about a year's delay getting in. Or is it a combination of both, or is it, what do you think that is?
The politically correct answer, I'd say it's a combination of all of those things. But I think people can draw their own conclusions as to why there's been some real challenges in the US market and the development of and approval of new drugs.
Is that an opportunity for us?
It certainly is.
Absolutely.
Like there's people keen for stability. So.
Hello. So finally, I have access. So hello, folks. Brenton and Felicia, great talks. Thank you so much. Brenton, I love your vision. And of course, you know, we strongly support you at ARMI, the Australian Regen Med Institute out at Monash. Felicia, I'm really, really keen to come and have a chat with you because we are right next door. But I'm very interested to have an understanding how Moderna enables and grows and collaborates with startups and biotechs in the sector. I know a lot of corporates do this, and really interested to have an understanding of how you do that. You kind of indicated that academics can apply and access RNA and lipid nanoparticles, but just really interested to understand the biotech story as well.
Yeah, so it's evolving here in Australia, and a lot of the sort of the I suppose small to medium-sized enterprises that we work with are actually more in the artificial intelligence and quantum computing space at this point in time, so that's, you know, from an Australia perspective, but there have been sort of strategic relationships that Moderna Global have had with various different SMEs and small startups. So look, Manny, I'm happy to have a conversation with you and see what you're thinking and talk to you about, you know, what opportunities there might be for collaboration on some projects.
I'd love to. Thank you so much. There's quite a bit to chat. Brenton, love your vision and keep pushing on. We will advocate with you.
Thanks, Maggie.
So we've got one more question here in the room and then we'll wrap.
So first, thank you. Both really interesting and very informative presentations. Can we get the slides, please? And then I have a substantive question. So for Felicia, how much of a prompt for setting up the facility was the pandemic, or were you already in discussions with the Victorian government or the federal government about doing some kind of regional centre before that happened?
So Moderna's always had an interest, you know, more widely, but it was actually a Commonwealth tender. So the Commonwealth put out a tender because they wanted mRNA capability, and so multiple different organisations applied and made a submission to that tender. And Moderna ended up being the successful applicant. So it was a fair and transparent process that was undertaken. And that was with the Commonwealth, and then there was a competitive process across the states because they wanted Moderna to go to the various different states, and in the end the Victorian submission was the one that, you know, made the most sense for us to come here. But I will, you know, I really want to emphasise though, Moderna really is focused really focused on Australian-wide integration of capability, not just Victoria. And I mean, we are really strong here in Victoria, but there are some amazingly smart people across the country that have some really excellent expertise. And so, you know, we work really collaboratively, and there are other facilities like the base facility up in Queensland, which is really good for academics to get access to mRNA and LNP, and and so we're really sort of cognizant and sort of working together, you know, to grow the Australian ecosystem. And as I say, is that co-opetition, healthy co-opetition. Yeah.
But the pandemic was the trigger.
It was, the pandemic was absolutely the trigger for the tender that the Commonwealth set, yeah.
So there had been no visionary discussions in the whole Victorian government, but we must have.
I don't know what happened in the Victorian government beforehand, and so I really don't know what happened within the Commonwealth and Victorian governments beforehand. But that was the trigger for Moderna to put the application through, and I think Moderna in itself, of course, we had the site in the United States, but also recognition that the capability has applicability across the globe. And so they were very much about, what are the relationships that we need to stand up as an organisation to support, not only public health, but, more broadly, the various different portfolios. So I think it was just circumstances, timing, that it all sort of converged at that point.
Just add one other thing is that it's amazing that we could stand this up, the Moderna facility, in 4 years. It's just amazing. When was the last time we stood up an end-to-end drug production facility? It's not because we couldn't do it, we just chose not to do it. Now that we've done it again, like there was a CSL, that was years and years ago, started off as Commonwealth Serum Laboratories. Massive gap in time to Moderna. What's next? What's stopping us from doing the next? And I think, that's the question that maybe should be asked is, why can't we have a proper pharmaceutical industry in Australia that's doing this end-to-end production? They're producing high-value, low-volume products. Tyranny of distance doesn't necessarily apply. Shipping high-value products. And I think it's a demonstration through Moderna. We can do it if we want to do it. So, put the question forward.
Brendan, I think you've landed right in exactly where we need to be to wind up our event tonight. Something that you said quite early in your presentation was start with the end in mind. And sad to say, of course, it was a global pandemic that had us all thinking about what we needed, and our the foresight of the governments, both here in Victoria and federally, where we now do have shared infrastructure. And the stories that we've heard tonight about what's required to have efficient, effective, safe, end-to-end biomanufacturing, and then what has been demonstrated feasible on what I've been reminded was the old Clayton Drive-in site. It's just extraordinary. And the fact that what we can look at now is prevention. We have some preparedness, and I think the sharing of that infrastructure is so important. The sharing of the knowledge, the investment in more capability, both within Australia, but also putting Australia in a leadership position, perhaps the one that it ought to have in our region, and perhaps most importantly, the investment in the talent. The development of an immersion centre for training that's very specific but has flexibility, that is built and can riff off all that's been achieved in these last 4 years out of what certainly in my lifetime has been a most unfortunate event to have lived through. Fortunately, we have. I want to thank both of our speakers tonight for the preparation that they put to their talks, to the way that they've complemented one another in their presentations, the thoughtfulness of having worked together and to have shown that through in their presentations as well, so that we as an audience, both here in the room and online, have had, I think, one of the most fascinating journeys through the whole concept of biomanufacturing and the multitude of elements that entails. But the power of its achievement and the fact that when we work together we can, there's not that many barriers to success when we put all of our bright minds together. So join with me, please, in thanking Brenton and Felicia for some fabulous work tonight.